What many people might be surprised to know is that in a whole variety of diseases, ranging from neuropsychiatric diseases like depression, schizophrenia, to neurodegenerative conditions like Alzheimer's disease and Parkinson's disease, there is physical atrophy of this brain region. Synapses are lost, dendritic spines are retracted, and the branches physically pull back.
Welcome to Emery Pharma Speaker Series. Today we have Dr. David Olson, Associate Professor in the Department of Chemistry and Biochemistry and Molecular Medicine at UC Davis. He holds affiliate appointments with the Center for Mind and Brain and the Center for Neuroscience and serves as the Chief Innovation Officer and Head of Scientific Advisory Board at Therapeutics, a company he co-founded. Dr. Olson is a leading figure in the central nervous system drug discovery space, best known for his work on psychoplastogens, a class of compounds that enhance neuroplasticity and have therapeutic potential for depression, PTSD, addiction, and neurodegenerative diseases. He is also the first ever recipient of the Dr. Mohsen Najafi Award in medicinal chemistry. Dr. Najafi was my grandfather who passed away in 2019 from dementia. The research you'll hear about today is a powerful reflection of why my family established this award and continues to support the chemistry research at UC Davis. Dr. Olson earned his undergraduate degree from Union College and completed his PhD at Stanford University. He also conducted his postdoc research at the Broad Institute of MIT and Harvard, and then joined UC Davis faculty in 2015. We're honored to have him here today, so please give a warm welcome to Dr. Olson.
Hi, everybody. It's great to be with you today. I'm just going to share my screen. Well, it's great to tell you all a little bit about the work that we've been doing here at UC Davis. But before we get started, I need to tell you that in addition to my academic appointments, I'm also a co-founder and the Chief Innovation Officer of Therapeutics. But everything I'm going to tell you about today is work done in my academic laboratory at the Institute for Psychedelics and Neurotherapeutics at UC Davis. To understand our approach to treating a variety of different brain conditions, you need to know something about the structure of neurons in a part of the brain called the prefrontal cortex. It's right about here. What I'm showing you now is the structure of a pyramidal neuron in the prefrontal cortex in a healthy individual. You can see that this neuron has lots of these branches, dendritic branches, and if you zoom in on one of those branches, you'll see these little tiny nubs called dendritic spines. Now, these spines are the sites of excitatory synapses in the brain, the way that two neurons will communicate with each other. What many people might be surprised to know is that in a whole variety of diseases, ranging from neuropsychiatric diseases like depression, schizophrenia, to neurodegenerative conditions like Alzheimer's disease and Parkinson's disease, there is physical atrophy of this brain region. Synapses are lost, dendritic spines are retracted, and the branches physically pull back. And so our approach really has been to try to find small molecule drugs that can restore these lost synapses and repair this damaged circuitry. And we thought that if we could do that, we might be able to create therapeutics that are broadly applicable. And the reason is because the prefrontal cortex is a central hub in the brain. Some people call it the conductor of the brain because it projects to a whole bunch of other regions that do things like regulate drug-seeking behavior and reward, control the expression of fear memory, regulate motivation, et cetera, et cetera. And so it's a pretty critical brain region. And so many years ago, we started studying a class of drugs that we call psychoplastogens. And these are just chemical stimuli that are very good at promoting the growth of neurons within the prefrontal cortex. They induce neuritogenesis, the growth of these new neurites. They induce spinogenesis, the growth of those dendritic spines. And these structural changes are often accompanied by functional changes that you can measure with traditional techniques like electrophysiology. Now, when we were looking for classes of compounds that might serve as psychoplastogens, we're really drawn to the classic serotonergic psychedelics like LSD and psilocin, as well as kind of the atypical psychedelics like MDMA and ibogaine. And what our group found many years ago, way back in 2018 is when we reported this, is that all of these drugs are very good at promoting cortical neuron growth. And so what I'm showing you here are some neurons that were treated with the vehicle control in the middle versus the psychedelics, DMT and LSD on the left and the right. And you can see very clearly that the neurons treated with psychedelics have more complex dendritic arbors. And if you zoom in on a dendrite and do some super resolution microscopy, you can see more of these dendritic spine-like structures and these filopodia-like structures. So to demonstrate this in vivo, we relied on using a psychedelic that is rapidly cleared from the body, dimethyltryptamine or DMT. The half-life of DMT in a rodent is only about 10 minutes or so, 15 minutes. And so for this experiment, we gave a single IP injection of DMT to a rat. We then waited 24 hours, a time point when all of the drug has been cleared from the body. We then sacrifice the animal, take tissue, look at structure via Golgi staining, and measure function via electrophysiology. And what we find is that even long after the drug has been cleared from the body, we see a large increase in dendritic spine growth in the cortex. And that's accompanied by these functional effects, increases in both the amplitude and the frequency of these spontaneous excitatory postsynaptic currents. So next, we really wanted to understand what was the mechanism by which psychedelics promote this type of neuroplasticity. So we started doing some selective pharmacology. One of the first targets that we looked at was a particular serotonin receptor called the serotonin 2A receptor, or 5-HT2A receptor. And the reason we were interested in that is because it's well known that the 5-HT2A receptor mediates the hallucinogenic effects of psychedelics. And we found that if we treated with a 5-HT2 antagonist, we could block the effects of psychedelics from 3 distinct chemical scaffolds: ergolines like LSD, tryptamines like DMT, and amphetamines like DOI. We also looked at a couple of downstream kinases that were implicated in compound-induced neuroplasticity, in particular ketamine-induced neuroplasticity. One is a kinase called TrkB, which is the high-affinity receptor for brain-derived neurotrophic factor. Another one is this kinase called mTOR, which is responsible for the production of all the proteins necessary for plasticity, all of the structural proteins that allow neurons to change their shape, and all of the ion channels that allow them to change their synaptic weights. And if we block TrkB or mTOR, we completely block the effects of psychedelics on neuroplasticity. So, next, we wanted to be even more convinced that this was the pathway that we were targeting. This particular antagonist that we were using for the serotonin 2A receptor is not a perfectly selective compound. It hits a couple of other receptors. So we wanted to rely on genetic models. And so for these experiments, we used wild-type animals and these 5-HT2A knockout animals, and we treated with the psychedelic dimethyltryptamine. And we found that in vivo, there was a very large increase in spine density in the wild-type animals, and that effect was completely gone in the knockout animals. Moreover, the functional effects were also completely gone in the knockout animals, really suggesting that the 5-HT2A receptor was a key mediator of psychedelic-induced neuroplasticity. Now, that was all fine, but we had one particular result that really kind of bothered us for a long time, and it was this one. We noticed that this compound over here on the left with the hydroxyl group in this position could not promote cortical neuron growth in the same way that a psychedelic could. And that bothered us because that molecule is serotonin. And I just told you that the serotonin 2A receptor was responsible for psychedelic-induced neuronal growth. We know that serotonin is a very good activator of that receptor, so it didn't really make a lot of sense to us why serotonin couldn't do the same thing as a psychedelic. We really didn't fully understand this until we saw this particular structure-activity relationship study. So we were taking these very polar compounds like serotonin and simply adding a little more grease to them. We added 1 methyl group, then we added 2 methyl groups, and we found that as you increased methylation, you started to produce compounds that were better at promoting cortical neuron growth. So the next thing we did is we tried to correlate this cortical neuron growth phenotype with a standard measure of 5-HT2A receptor activation, and we really don't see any correlation at all. But then when we try to correlate the cortical neuron growth phenotype with the lipophilicity of the drugs, we see something really interesting. The very polar compounds were very bad at promoting growth, but the non-polar psychedelics were very good at promoting cortical neuron growth. And that gave us the hypothesis or the idea that maybe the reason that serotonin could not do the same thing as a psychedelic is because maybe it can't get to the target. Maybe this particular G protein coupled receptor is not exerting its effects from the plasma membrane. Maybe there is an intracellular population that is more important for these plasticity promoting effects. And so to answer that question, the very first thing that we did is we just looked for where the receptor was expressed. This is a non-neuronal cell line. And when you express the 5-HT2A receptor in a non-neuronal cell type like a HEK cell, you see the typical GPCR expression pattern with a lot of it on the cell surface. But when you look in a cortical neuron, you get something totally different. Now you see that most of the receptors are localized inside the cell, and they overlap really well with a marker of the Golgi complex. So next, we did something that I think only a chemist would think to do, is we took these membrane permeable compounds, 2,5-HT2, an agonist, DMT and psilocin, and this 5-HT2 antagonist, ketanserin, and we converted them into membrane impermeable versions of themselves by giving them a permanent charge. We then run our experiments under two different conditions. One is just plain old neurons, and the other one is with a little bit of electroporation. So with electroporation, you can transiently apply a voltage to poke holes in the cell. That allows the impermeable compounds to cross the membrane, the holes close, and then the impermeable compounds get stuck on the inside of the cells. And when we do this, we find something really interesting. Without electroporation, we get the results that we would expect. The permeable agonist DMT and psilocin promote growth. The impermeable agonist TMT and psilocybin do not. Similarly, the permeable antagonist can block psychedelic-induced growth, but the impermeable antagonist cannot. But when you electroporate, you kind of flip that around. So now the impermeable agonist TMT and psilocybin can promote growth, and the impermeable antagonist can block growth. So that really suggested that it was an intracellular population that was responsible for this. Now, if that was true, we reason that serotonin should be able to promote growth just like a psychedelic if you could just get it to the target. And so for these experiments, we relied on a particular transporter of serotonin called SERT. We took cortical neurons and we sparsely transfected them with SERT. So some neurons, like this one on the left, have the transporter, and some neurons, like the one on the right, do not. So then when we run this study, we can compare the two different genotypes, SERT negative and SERT positive. We find that the vehicle-treated neurons, there's no growth. The DMT-treated neurons, both genotypes grow because presumably DMT can just passively diffuse across the membrane and activate the receptor. But serotonin only promotes growth in the SERT positive neurons, the neurons capable of importing serotonin. So next, we replicated this study and extended it a little bit. So this first part right here is just a replication of what you saw on that last slide. But now we added the 5-HT2 antagonist ketanserin. And when you do that, you see that you completely block the serotonin effect. And we also added this drug citalopram, which is a selective serotonin reuptake inhibitor. It blocks the SERT transporter. And when you use citalopram, you also block the effects of serotonin. So that suggests that serotonin needs to be transported by SERT and then activate the 2A receptor in order to exert its effects. In contrast, DMT promotes growth in both genotypes. Ketanserin blocks the effects of DMT in both genotypes, but citalopram cannot block the effects, and that is presumably because DMT does not require transport by SERT. It can just passively cross the membrane. So all of these were neuritogenesis assays. We also ran spinogenesis assays, and we see very similar results. Again, DMT promotes growth in both genotypes. Serotonin only promotes growth in the neurons capable of importing serotonin. So next, we wanted to test this in vivo. And so the way we did this was to use a virus to express the transporter on the postsynaptic side. Normally, the serotonin transporter is expressed on presynaptic terminals that spit out serotonin so that it can reuptake serotonin into those nerve terminals. But we expressed the transporter on the postsynaptic side, the cortical neurons that we're looking to promote the growth of. And so after a few weeks to allow for expression of the transporter on the postsynaptic side, We then give this drug parachloroamphetamine, which is a selective serotonin releasing agent. And then 24 hours later, after the drug has been cleared, we come back and we look at dendritic spine density. And what we find is that in the neurons capable of importing serotonin, we see a nice increase in dendritic spine density. We reason that if this effect on cortical neuron growth was responsible for the antidepressant effects of psychedelics, then we should see a similar type of antidepressant response in a behavioral assay. And so this experiment was basically exactly the same as what I just showed you, but we first ran a novelty-induced locomotion assay to just demonstrate that there's no difference between the genotypes. There wasn't. We then run this forced swim test as a measure of antidepressant response. The first forced swim test, it really just provides some stressor to the animals, and it also ensures that the two genotypes are evenly balanced. They are. We then gave the parachloroamphetamine. 24 hours later, came back and did the test of efficacy. And what you see is that in only the animals that are capable of importing serotonin into those prefrontal cortical neurons, those are the animals that have an antidepressant response. So at this point, we're feeling pretty good that the plasticity promoting effects and at least some of the antidepressant effects of psychedelics are mediated by this intracellular population of 5-HT2A receptors. But there's a problem. And the problem is that these are hallucinogenic compounds, and that dramatically limits their clinical scalability. In order for psychedelics to be administered safely and effectively, they need to be given in the clinic, usually under the supervision of multiple healthcare professionals, which increases the cost and the complexity of the treatment and limits the total number of people who could really benefit. And so we wondered if it would be possible to separate the hallucinogenic effects of psychedelics from their beneficial effects on cortical neuron growth. And we tried to address this problem in a variety of different ways. One way that we did this, we recently published with Tina Kim, was using a really interesting genetic construct that allowed us to tag psychedelic responsive neurons in the prefrontal cortex and then come back later and reactivate them with optogenetic stimulation. And what we found is that when we reactivated those neurons later, we drove a therapeutic response, but not a hallucinogenic response. That told us that the circuitry is distinct. And that told us that it would be possible to activate one circuit without activating the other. Really important from a basic science perspective, very difficult from a drug strategy if you want to actually treat a patient. So we started thinking about ways we could use drugs to achieve a similar type of dissociation of the therapeutic and hallucinogenic effects. And the first thing that we tried was to simply use subhallucinogenic doses. And we found that even subhallucinogenic doses produced beneficial effects on depression and anxiety-related behaviors. But the problem with this approach is that there's still abuse potential. Patients could simply hoard pills, take a whole bunch at the same time to achieve a high hallucinogenic dose. So what we wanted to do was to engineer completely novel chemicals that do not produce a hallucinogenic effect at any dose. And for that, we really needed a very predictive assay of hallucinogenic potential in people. And so we turned to this behavioral assay. It's called the mouse head twitch response. You can see the behavior in the lower left-hand corner. It's this rapid rotational movement of the head. And Adam Halberstadt and coworkers have done some really beautiful work demonstrating that human hallucinogenic potency correlates exceptionally well with the rodent head twitch response. And so this is a really important assay for us. But the problem is it's an in vivo assay, and so it's hard to screen lots and lots of compounds. So we needed a cellular assay in order to do drug discovery. When we started this work, we really didn't have any options available to us. And so the way we solved this problem was actually pretty simple. We knew that the serotonin 2A receptor was responsible for the hallucinogenic effects of psychedelics. So we simply took that receptor, we lopped off the third intracellular loop, and we fused on a circularly permuted green fluorescent protein. And the way this technology works is that there's basically a slit in the beta barrel that allows the fluorescence to be quenched of the fluorophore. When a ligand binds to the receptor, that induces a conformational change that closes the beta barrel. that allows fluorescence to turn on. You can see that these antagonists either do nothing or turn off fluorescence. And we got very excited when we found that potency in this biosensor assay correlated very nicely with human hallucinogenic potency. So we started calling this biosensor Psychlite. And I would say we were even more excited when we found that Psychlite could differentiate between a hallucinogen and a very closely related structural analog. And so here is a very potent hallucinogen LSD and this compound glyceride, which is non-hallucinogenic and has actually been used to treat Parkinson's disease. You can see that structurally they're almost identical. The only difference is glyceride has this nitrogen here and that stereo center is just swapped. Now you might be asking yourself, well, maybe glyceride doesn't actually bind to the biosensor. So to answer that question, we just run the assay in antagonist mode. We give a little bit of agonist to turn it on, and then we titrate in increasing quantities of the non-hallucinogenic ligand, and we watch the fluorescence decrease. So we were able to run this assay in agonist mode on the x-axis and antagonist mode on the y-axis. I'm going to point your eye to this little black dot. That's the vehicle control for both. And what we find is that compounds that increase Psychlite fluorescence more than about 1 standard deviation in agonist mode, these are all known hallucinogens. Compounds that decrease Psychlite fluorescence more than 1 standard deviation in antagonist mode, these are all non-hallucinogenic ligands. And you can see some really interesting compounds here. You see traditional antagonists like volanesorine and ketanserin. You also see antipsychotics like haloperidol, but you also see these non-hallucinogenic 5-HT2A partial agonists like glyceride. And we use this to actually identify this compound that we call AAZ as a potential non-hallucinogenic psychoplastogen. In our biosensor assays, it predicted that AAZ was going to be non-hallucinogenic. And in our head twitch response assays, we found absolutely no head twitches, even up to a massive dose of 100 mg per kg. Therapeutic dose of this compound is closer to 10 mg per kg. And we found that it had very comparable psychoplastogenic effects as ketamine. So here's just a concentration response. It produces both rapid and sustained antidepressant effects comparable to ketamine. And here's an example of a depression assay that measures anhedonia. We use these VMAT2 heterozygous mice developed by Bill Wetzel's group. These mice have some baseline anhedonia. They don't prefer sucrose solutions like typical wild-type mice. You can see that that deficit here. The wild-type mice prefer the sucrose. VMAT2 heterozygous mice do not. A single dose of AAZ can rapidly rescue this anhedonia phenotype, and the effect persists. It persists for at least a couple of weeks before the genotypes start to diverge again. We've been using Psychlite and a whole bunch of other tools to do drug discovery on every single major psychedelic scaffold, tryptamines like psilocin, amphetamines like MDMA, ergolines like LSD. But what I want to tell you about now is our efforts to work on analogs of this compound right here, ibogaine. Ibogaine is what you might call an atypical psychedelic, but it has some really interesting properties. There are anecdotal reports and some open label clinical trials that suggest that a single dose of ibogaine can, keep people suffering from opioid use disorder drug-free for up to 6 months. And a second dose can keep them drug-free for up to 3 years, which is just remarkable. And so you might be asking yourself, well, why aren't we using, ibogaine to address the opioid epidemic in this country? And the answer is that ibogaine is a pretty terrible drug for a variety of reasons. Number one, it's really difficult to synthesize. When we started this work, there were no de novo total syntheses that could produce it enantioselectively or in quantities sufficient for clinical use. Number two, it's a rock. It is very insoluble in water, which makes formulation quite challenging. Of course, it has these hallucinogenic effects that limit its therapeutic potential. But the worst thing about it is that it binds to hERG channels in the heart and can cause cardiac arrhythmias. And several people have actually died when taking ibogaine to treat their opioid use disorder. So we wanted to solve all these problems. So we can start with the simplest one, de novo total synthesis. And I'm not going to go into details here. You can read about it in this Nature Chemistry paper that just came out this past year. We developed a more efficient synthesis of ibogaine. So for now, maybe let's just call that problem solved. But we haven't solved the other problems. And no matter how much ibogaine you can produce, you're always going to have these issues remaining. The only way to solve those problems is to come up with a new compound. And so for that, we really relied on something called function-oriented synthesis. It is a technique pioneered by people like Paul Wender. And the idea is pretty simple. You take a complex natural product like ibogaine and you just distill it down into the structural features that give rise to the function that you care about. In our case, that cortical neuron growth phenotype. And so if you look at the structure of ibogaine, you can see it's got this indole ring, it's got this 7-membered azapine ring, it's also got this bicyclic isoquinoline. And so all we did is we simply started deleting bonds. If you delete the bond between the indole and the isoquinoline, you make compounds like scaffold 1. If you delete the isoquinoline altogether, you make compounds like scaffold 2. Now, I'm not going to take you through all of the medicinal chemistry, but I'll just show you a few of the highlights. What we found was that compounds that contained the tetrahydroazapine but lacked the isoquinoline, well, they were pretty good at promoting cortical neuron growth. But the opposite wasn't true. If the compound lacked the azapine, they were pretty bad for the most part. So we decided to prioritize this compound. The structure of that compound is exactly the same as ibogaine, just lacking the isoquinoline. So we started calling this compound ibogainalog, or IBG for short. We made one other small structural modification. We just took the methoxy group, moved it from the 5 to the 6 position to make this compound, which is very similar to the natural product tabernanthine. So we started calling this tabernanthalog, or TBG. Now, unlike ibogaine, TBG can be produced in a single step using this Fischer-Indole cyclization in gram quantities. It also has a much better safety profile. So here's some immobilized zebrafish. You can see the bradycardia that ibogaine induces. You can see the change in the hERG affinity. So here is ibogaine's hERG affinity. TBG is about 150 times less potent for hERG inhibition than ibogaine is. It also lacks 5-HT2B agonism, which is a problem that plagues many psychedelics, including 5-methoxydimethyltryptamine. You can see that TBG is actually an antagonist at 5-HT2B receptors. And it produces absolutely no head twitch response and actually can block a psychedelic-induced head twitch response. So this all suggests that it's a safer analog of a psychedelic. But none of this matters if it doesn't produce ibogaine-like efficacy. And so the next thing that we did is we did some 2-photon imaging using mice that have neurons labeled with a fluorescent protein in their prefrontal cortex. This was in their somatosensory cortex. And for this experiment, we created a window in the skull. We used a 2-photon microscope to peer into the brain, look at a section of dendrite, give tabernanthalog, come back 24 hours later after the drug has been cleared, look at the exact same section of dendrite. And when you do that, you can see that some spines are formed, like this one here in blue, and some spines are eliminated, like this one here in red. We found that TBG really didn't affect spine elimination rate at all, but it did increase spine formation, and it did so to a comparable extent as the hallucinogenic drug DOI. Now, these changes in structural plasticity are accompanied by beneficial behavioral effects, including antidepressant-like effects in the forced swim test, anti-addictive effects in this model of alcohol use disorder, and anti-addictive effects in this model of heroin use disorder. So next, we wanted to understand the mechanism a little better. Tabernanthalog is a non-hallucinogenic compound, so it's possible that it was working through a completely different receptor than classic serotonergic psychedelics. But we found that that wasn't the case. We use these 5-HT2A knockout animals, and just like with psychedelics, there's a big effect in the wild-type animals. The effect is completely gone in the 5-HT2A knockout animals. Here is another behavioral test of depression. Again, you see a nice antidepressant response in the wild type, no effect in the knockouts. So we have good evidence that the 5-HT2A receptor is mediating the plasticity promoting effects of of TBG, as well as some of the antidepressant effects. But I haven't shown you data that links the two. And for this experiment, we teamed up with Connor Liston at Weill Cornell, and his lab uses this really interesting construct. It is this photoactivatable RAC1 that is targeted to activated synapses. The way this experiment works is we give TBG, we wait 12 hours for the dendritic spines to form, We then photoablate specifically the TBG-induced dendritic spines, and then we look for antidepressant results. And we found that when you photoablate those spines, the antidepressant effects of TBG go away, suggesting that structural plasticity in the cortex is required for the sustained antidepressant effects of that drug. So next, we wanted to compare the efficacy of TBG and its closely related psychedelic analog, 5-methoxy-DMT. We found that they had equivalent effects on spine density and on EPSE frequency and amplitude. We next wanted to see what do they do acutely to the brain. We know that the 5-HT2A receptor is a GQ-coupled receptor that regulates intracellular calcium levels. And so for this experiment, we expressed a sensor for calcium in the prefrontal cortex. We then did some 2-photon imaging to see what happens when we give the drug. And we were able to identify specific cells and track them before and after drug treatment. And what we find is pretty interesting. We found that 5-MeO and TBG activate roughly the same number of cells, inactivate roughly the same number of cells, and then there's some cells that just don't respond to either of them. So they produce very comparable effects on intracellular calcium in the cortex. But we wanted to know, what do they do to glutamate levels? It's known that with psychedelics, they can cause this glutamate burst, and that was believed to underlie psychedelic-induced neuroplasticity. So for this experiment, we used a biosensor for glutamate, again, putting it in the prefrontal cortex. And we found that a single administration of 5-MeO-DMT causes this bursting of these glutamate transients. You don't see this with TBG at all. And we wondered what this would do to immediate early gene activation. There's been a lot of reports that psychedelics promote immediate early gene activation, and people have linked that to their plasticity effects. But given that TBG caused comparable plasticity effects as 5-MeO-DMT, but caused no effects on glutamate, we wondered what happens with IEGs. And what we found is this. So this is some brains that had been cleared and imaged with light sheet microscopy, looking at CFOS expression. We found massive increases in CFOS expression across the brain with 5-MeO-DMT. We found absolutely no changes with TBG. When you look at just typical immunohistochemistry, you can see that again, lots of CFOS in the cortex after 5-MeO, nothing with TBG. Here's just another slice where you can see the somatosensory cortex really lights up with 5-MeO. Again, nothing with TBG. Now, that's all immunohistochemistry at the protein level. We also wanted to look at the mRNA level. So we did some single nucleus RNA-seq experiments. That allowed us to identify all of these different population of cells in the prefrontal cortex and rank them based on their 5-HT2A expression levels, and then look at their IEG expressions. And the takeaway is this, that 5-MeO-DMT, and I'm just showing one particular population here, causes a massive IEG response. You see FOS that I showed you a second ago with the protein levels, but you see other IEG responses as well. With TBG, you get nothing. There's absolutely no IEG response, about an hour or two after compound administration. So this really demonstrates that the mechanism of these non-hallucinogenic psychoplastogens shares some features with psychedelics in that it involves 5-HT2A activation, but they are distinct, that there are some additional things that accompany the psychedelic response. So finally, I wanted to end by talking a little more about our efforts to take these non-hallucinogenic psychoplastogens into patient populations where psychedelics might be contraindicated, like patients with schizophrenia or dementia or psychosis and neurodegeneration, these kind of things. And a lot of the work I'm going to tell you about now was generously supported by the Najafi family through a grant. Here, what I'm showing you is neurons in the prefrontal cortex from patients with schizophrenia. These are post-mortem samples, and you can clearly see a loss of dendritic spines in this disease. And this is one of the hallmarks of schizophrenia, and it's believed to underlie not only the positive aspects of the disease, like the hallucinations, but also the negative aspects like the anhedonia, and also the cognitive symptoms of the disease. We thought that if we had a drug that could promote the growth of these neurons, but could remain non-hallucinogenic, we might have a very, very powerful treatment for schizophrenia and related conditions. So we started thinking about, well, what is the psychedelic that has the largest effects on dendritic spine density and is one of the most potent psychoplastogens we've ever studied? And the answer was LSD. So we were wondering, could we take LSD and engineer it to be non-hallucinogenic? And fortunately, we had some really nice structural information from a variety of different groups. And in those structures, we realized that LSD makes a very critical hydrogen bond interaction with this particular serine residue. And so we thought that if we just disrupted this one interaction, maybe we would make a non-hallucinogenic LSD analog. And so we thought this might be possible because we played this trick before with DMT. So we published this way back in 2020. We took DMT and we just flipped the indole upside down to make this iso-DMT scaffold. These two compounds are almost identical. They have the same overall shape and they have the same molecular weights. The only difference is the iso-DMTs cannot engage in hydrogen bond donation through the indole NH. So we just wanted to make this same exact modification to LSD. We started calling this molecule JRT. It was named after the graduate student Jeremy R. Tuck, who first synthesized it. Now, unlike LSD, which is created via semi-synthesis from lysergic acid, we would need to create a de novo total synthesis to make JRT. Before we started those efforts, we first did some modeling to see how might JRT bind to the 5-HT2A receptor. What I'm showing you here is the crystal structure of LSD bound to the 2A receptor. You can see that the indole nitrogen is very close to the serine residue. And when you dock in JRT, you basically get almost the exact same pose, with the exception that the indole moves about an angstrom away from that serine residue. If you look down on the receptor, the two structures are almost overlapping. And so that really suggested that there might be enough of a subtle difference in how JRT bound the receptor to change its pharmacology. So to make JRT, we first started with this commercially available nicotinic acid derivative. We then activate the acid and couple to make the diethylamide. We then do this SNAR reaction to forge that carbon-carbon bond. We do this pretty nifty oxidation, hydrolysis reaction, which converts this to the carboxylic acid. This can then be esterified to make the methyl ester. We then can do selective reduction of the ester in the presence of the amide to make this carbonyl compound. And then we can methylate the pyridine, reduce it to the tetrahydropyridine before doing a cross-coupling reaction to form that carbon-carbon bond. Finally, we activate this alcohol with tosyl chloride, and then in situ, we perform an SN2 reaction to displace the tosylate to make this carbon-nitrogen bond and create JRT in only 9 steps and 16% overall yield on gram scale. Now, when we tested JRT, we found something pretty interesting. LSD is a pretty promiscuous compound. It hits many, many neuroreceptors, including serotonin receptors. But JRT is a much, much more selective compound. It pretty much only has affinity for serotonin receptors. Moreover, it seems to be a partial agonist of 5-HT2A receptors. So here is LSD's effect on the human 5-HT2A receptor. Here is JRT's effect. You can see that it is a lower efficacy compound. Now, that serine residue we thought might be explaining the difference in efficacy between LSD and JRT. So we simply made this mutant where we mutated the serine to an alanine. And when you do that, you see a big difference in the activation of LSD. You see no difference in the activation of JRT, suggesting that our hypothesis was correct. In terms of hallucinogenic potential, JRT doesn't really have any of the telltale signs of a hallucinogen. It doesn't increase a locomotion or cause any psychostimulant-like properties. It doesn't have any effect on prepulse inhibition like LSD and other psychedelics do. It doesn't really produce much in terms of a head twitch response and can actually block an LSD-induced head twitch, but it produced very profound effects on neuroplasticity. Here is JRT compared to LSD and clozapine, the state-of-the-art antipsychotic compound, and it outperforms all of them in vitro. So then when we looked in vivo, we did some electron microscopy with Uri Manor at UC San Diego, and we found that JRT was very good at promoting spinogenesis and synaptic density in layer 1 of the cortex. Now, patients with schizophrenia tend to have a lot of atrophy in layer 2, 3, so we also look there. These are those mice that have their neurons labeled with that fluorescent protein. We can give chronic corticosterone to induce atrophy. You can see the atrophy here, but when we give JRT, we see a complete rescue of that atrophy. To look at the positive symptoms of JRT, we looked at a very traditional assay of dopaminergic function. This is amphetamine-induced hyperlocomotion. Now, a lot of antipsychotics have affinity for dopamine receptors, but that can lead to a lot of off-target side effects. It turns out that JRT is a potent agonist of 5-HT2C receptors, and that can indirectly modulate dopamine release into the striatum. And so we thought that this might impact amphetamine-induced hyperlocomotion without directly antagonizing dopamine receptors, and that turned out to be right. We see this blunting of this amphetamine-induced hyperlocomotion phenotype. We also wanted to look at the negative symptoms in schizophrenia, the depression. And so in a forced swim test assay and also the sucrose preference test assay, we can see we get really nice rescue of the behavioral deficits with a single dose of JRT. And you can see how potent it is. This is the dose in the forced swim test. We use ketamine, 10 mg per kg, as a positive control. That is the lowest dose of ketamine that you can get to produce antidepressant-like effects in these rats. But we can get antidepressant effects with JRT even down to 0.1 mg per kg, so suggesting it's at least 100 times more potent than the state-of-the-art fast-acting antidepressant. And then finally, we really wanted to look at the cognitive symptoms of the disease because this is something that current medications really don't do a great job at addressing. And so for this assay, we used something, a mouse behavioral assay that was analogous to a human test, something called the Wisconsin card sorting task. And the way that task works is you can have patients sort cards based on either number or color or shape, and patients will learn a rule for sorting the cards pretty well. And we can do the same type of test in a mice, and you can see that these 3 groups all perform equally well, just like patients and healthy controls in the clinic. But where patients really struggle is when you change the rule. When you don't tell them that you're changing the rule, but you just tell them that their responses are incorrect and they need to adapt. They need to adapt and figure out the new rule. Patients with schizophrenia tend to perseverate. They perseverate on the old rule and they have problems with so-called reversal learning. And here is an example where we use stress to induce deficits in this reversal learning, but we found that a single dose of JRT completely rescued those deficits, suggesting that it might impact the cognitive domain in schizophrenia. Now, we're super excited about all the early data with JRT. I showed you all the efficacy for assays relevant to schizophrenia. We believe that this drug may be effective for neurodegenerative conditions like Alzheimer's disease, and that's where our work is going now. So to summarize what I told you about today, I told you that in a healthy brain, neurons in the prefrontal cortex have a lot of branches. They have a lot of these dendritic spines. But in many, many brain conditions, including neuropsychiatric diseases like depression or schizophrenia, and neurodegenerative conditions like Alzheimer's disease, there is a physical atrophy of those neurons. Psychoplastogens, whether they're hallucinogenic drugs like psychedelics or non-hallucinogenic drugs like TBG, AAZ, and JRT, they can promote the growth of these key neurons and potentially produce therapeutic effects across a range of indications. And so in summary, I told you today that psychedelics promote neuroplasticity by activating that intracellular population of 5-HT2A receptors. I also told you that Psychlite is a novel biosensor that has the potential to predict hallucinogenic potential, or has the ability to predict hallucinogenic potential for 5-HT2A ligands. And I told you that TBG, AAZ, and JRT are potentially safer psychoplastogens. And with that, I just like to thank some of these key collaborators who helped in various aspects of this work, along with these funding agencies, including the Najafi Award in medicinal chemistry, that really fueled our efforts to get JRT off the ground. And we're very excited about where it's going. And then finally, I'd like to thank all of the incredible students I get to work with every day. I've highlighted the names of all of the students whose work appeared in this presentation. And lastly, I just want to thank you for your attention. And thanks to all of the supporters of the UC Davis Institute for Psychedelics and Neurotherapeutics. Yeah, there are a number of companies trying to develop these types of agents. The most advanced asset is actually a compound called DLX1 that is being developed by the company that I co-founded, Therapeutics. DLX1 has completed phase 1A and phase 1B studies. The phase 1B results have been presented at several conferences so far, and what they found is that the drug produces absolutely no hallucinogenic effects in people. They found a really interesting EEG biomarker suggesting it's engaging targets in the brain. And patients with major depressive disorder have a rapid response and a sustained response. And so they're gearing up to do phase 2 trials now.
And what's the target indication?
For that compound, that asset, it's major depressive disorder.
I have a question. So regarding the two compounds that we discussed today, when comparing them to SSRIs and other psychiatric medications, where oftentimes you see in the first few weeks of treatment lots of adverse effects and not a lot of therapeutic efficacy, would you say that you're seeing a similar profile in these compounds, or that you see efficacy, earlier on?
Yeah, this paradigm of using psychoplastogens is completely different than SSRIs. With SSRIs, like you said, you need to give the drug daily for a long period of time before you start seeing any efficacy. And because you're giving the drug daily, that often leads to a lot of off-target side effects. These drugs are not meant to be taken daily. They're not necessarily going to be a one-and-done treatment, but they're meant to be taken once a week, once a month, once a year, on an intermittent basis. And as a result, they are expected to have much lower side effects profiles than typical SSRIs. The other thing that's really unique about the drugs is that they work rapidly. It turns out that pretty much every single antidepressant treatment that we know of, be it a slow-acting antidepressant like an SSRI, be it a non-pharmacological treatment like ECT, TMS, exercise, acute sleep deprivation, they all promote the growth of those cortical neurons. The key difference with psychoplastogens is that they do it very robustly and rapidly. And so with an SSRI, you can see an increase in cortical neuron growth over time that correlates with its clinical efficacy, taking weeks to, several weeks to show anything, substantial. Whereas a psychoplastogen like ketamine or psychedelics, or maybe these non-hallucinogenic variants, can produce really large increases in cortical neuron growth within 24 hours.
David, did you see any cognitive improvement in any of your non-hallucinogenic? You might have alluded to some of that rat studies that you've done. Can you comment?
Yeah, so that we did a study in mice looking at cognition. Reversal learning is a classic cognitive test that is known to involve the prefrontal cortex. And it was a pretty profound effect on cognition I showed you the effect for JRT, but we also tested this with TBG as well. And we saw similar results with that non-hallucinogenic analog as well. So we're definitely affecting like mood domains but we're also affecting cognitive domains as well.
Great.
David, that was an excellent presentation. Thank you so much. Wanted to just point to a number of studies that are going on nicotinamide for glaucoma and some other neuropsychiatric disorders. And I was just wondering if you see any correlation between nicotinamide and any of these, plastogens, psychoplastogens that you're working on.
It's interesting. We haven't actually tested the effects of nicotinamide directly on cortical neuron growth, but those with a very discerning chemical eye will notice that, nicotinic acid is within the core structure of LSD. I don't know. I don't have a good answer for you, but it's very interesting.
And then if I could ask one more question about, we actually have patients with severe vision loss, oftentimes have visual hallucinations. So that's interesting that, we have hallucinations with other neuropsychiatric disorders. And so if we were to increase dendritic spine growth in, let's say, brain, could you correlate that to maybe the optic nerve, or can you actually kind of extrapolate that it would have a similar effect on the nerve, on the optic nerve?
Yeah, so this is a really important point, and this is an area that our work often gets misinterpreted. People think that if you take a psychedelic, you're getting like plasticity all over the place, and this is not true. One of the reasons we like this class of molecules is because of the expression pattern of the key receptors, in particular the 5-HT2A receptor. And so that is expressed on particular cell types in particular circuits that seem to mediate beneficial effects. If you promote plasticity in other circuits, you can actually cause adverse effects. Drugs of abuse will cause plasticity in the mesolimbic pathway, and that's not necessarily a good thing. There are other compounds that can potentiate nociceptive neurons and cause chronic pain. That's not necessarily a good thing. The other thing that we've noticed is that when you give a psychoplastogen to just a healthy wild-type animal, it's really hard to promote the growth of those neurons because the brain has homeostatic mechanisms to keep that in check. You don't want to have a whole bunch of new synapses or dendritic spines because that could lead to things like, seizures. But what we find is that in a neuropsychiatric disease where you have a deficit, you have a loss of dendritic spines, you can use a fairly low dose of the compounds to get complete rescue of those. And we've been able to do 2-photon imaging to see, are we making a whole bunch of new synapses, or are we just repairing the damaged ones? And it turns out that when you induce a deficit, you can actually watch the dendritic spine pull away. And then when you give the drug, more often than not, you're just reconstituting the broken synapse. You're not making a whole bunch of new synapses. So to get to your question about, the optic nerve, is it possible to use these types of molecules to heal other circuitry? I would say potentially. It all depends on the expression of particular receptors in those circuits that you care about. What we are doing now is trying to tailor these psychoplastogens for various circuits so they can be useful for one indication versus another. Some people are very interested in using them for, healing damaged neural circuitry in the periphery, even spinal cord injury and those kind of things. We've used them in other, non-CNS areas that, I'm not really ready to talk about now, but, we've been seeing some pretty promising results.
Thank you so much. Thanks, David. And let's give David another round of applause, and really appreciate you joining us. Look forward to seeing you at UC Davis soon.
Thank you all.
All right.
Thank you.
Thank you so much.
Thank you.